Good morning, everyone. My name is Filomain Nguemo. I'm coming from the University of Cologne, that's in Germany. First of all, I would like to thank the organizer for having invited me here the first time. I have been in Cambridge several times, but not for the Sofyan Congress. So thank you for that, and I appreciate to be here to share my experience and summarize some of our data that we generated a few years ago on the EU big consumption project, making of more than 70 research group, and yes, and we close it. So yes, I will just show some part of the data, and it is about the early identification of biomarkers that are involved in cardiovascular, cardiotoxicity. So as you may know, the aim is to develop a really in vitro approach methodology to capture the very first signal of the toxicity in different organs. So on this project, we have nearly 3 target organs: heart, liver, and kidney. But my group was just focused on cardiotoxicity. And to do that, of course, we have to understand the mechanisms, how toxicity happens. Toxicity will happen when we first start, the compound will bind to some specific initiate some event. So this will drive all this cascade until we have in the level of the cells that show, the cell will show a signal that we can capture maybe by electrophysiology tool or some. But before that, we have to go through some cascade that we have to capture before. So this is very important. It's why this guy from cosmetic industry and the AO put together more than 8.6 million in 5 years to develop this approach. So for these 3 organs. So focusing on the heart, of course, we have to choose the cells models. We have to define the right protocol. We have to define the precision, which tool can we use to really capture that event at every time point from the first time that we put the draw on the presence of the cells or different cells models until the end of the draw exposure. It can be the short time, can be the long time. We have to define how to proceed. So appropriate biological model, we chose stem cells because I'm also specially in stem cells, and we have to choose the endpoint that is qualitative and quantitative accessible, and we have to choose the test protocol. It can be short time, long time as mentioned, and the tool to capture. And for that, for the cardiotoxicity, we chose actually the doxorubicin. Most of you will know that it is effective anti-cancer drug. And it is, however, very toxic for the induced high toxicity in the many patients and generate some arrhythmia. And the mechanism of doxorubicin is clearly known. And within this project, we do not only choose doxorubicin as gold standard compound, we chose other compounds that are known to generate some kind of arrhythmia on the cardiac cells, affect the electrical activity. So to be as test of proof or proof of concept. So yes, as you can see here, the different mechanisms of action of the doxorubicin. I will not go in detail, but we go through using the omics data to generate the effect of this on the heart organ. So to choose the cells model, as I mentioned, we are working with stem cells. So we generate a lot of stem cells from mouse, from monkey, from different animals. And today, thanks to the discovery of from the lab of Yamanaka in Japan, so that we can, for each person taking blood, anything on your body, and we can go to the lab, make some kind of 3-factor, putting 3 factors together and generate some embryonic stem cell-like. So, and this is possible from this to generate all the kind of stuff we have on your body. So, for example, we can use it to study the differentiation aging beings. We can, for specific area, put some draw in the first day of differentiation, and we go through all the process to go to the heart or to the kidney or to the liver formation. Of course, through all the embryogenic development to tissue. We can use to understand and prevent any disease. We can also use to test draw to identify draw target like heart, and also to reduce the use of animal in research as well. But it's more than that. It's not only, not that there are some more things coming out, what stem cell can be used for. So, and for this, we chose the stem cells, means we have our cells that we derive in vitro, and then we bring this cell to cardiac cells, and we characterize them. So first, I don't know why it's not moving. First, you find here different type of cells that we have on our life. This is like blood pluripotent stem cell that we just put some specific cocktail for 14 days, and then we have cardiac cells. The cardiac cell is not just looking nice, they are beating, and you wonder why you have this green. The green has the advantage that because the purpose is to make a transplantation, if we transplant those cells in infection area of the mouse, we'll be able to see if this cell has been differentiated or fused together with the original cell. This is why we mark this cell with some specific protein. Yeah, don't wonder why it's green. And here it's a recently developed tool used from human, Brugada human iPSC diseases model that we show that they are really have some defect in the contraction structure. So this is just to prove that we generate the cardiac cells. And also, we have to be sure that they are really cardiac cells. We make some kind of staining with specific protein for cardiac, troponin T, using light chain, small muscle, actin, and prove that they are pure as cardiac cells. So it's not only the protein expression, they have to be function, and we dissociate those cells, this cluster, beating cluster, we have to have single cells, and you can see that one single cells beating, and we use, of course, electrophysiology to quantify, because just imagine we generate the cluster of the cells, and these cells are beating, they are beating, but in the heart, we have different cardiac subtypes. We have to define which subtype it is involved in this cluster. And the only way to do that, it is electrophysiology. There's no other specific marker for ventricle, for nodal or atrial. It's why we just have to use electrophysiology. And you can imagine how hard it is to do it with manual Pascal. So not only the action potential just recapitulate all the ion channels that are expressed in cardiac, but you have to go inside to measure different ion channels that are involved and to test the response to different draw. Beta receptor agonist and muscarinic antagonist as well to see if they are responding, if the cells are responding to this draw. Of course, we measure all these ion channels that are involved and to be confirmed that they are really expressed and also function. And here you can see clearly the T and the L-type calcium canal with that. And from there, you have different ion channel expressed. So for that, we are sure that we have a target, the good cells to do what toxicity test. Chose the doxorubin as compound. We define the concentration. We put it in the presence of these cells and different time point, single dose exposure or different dose exposure, and we prepare different samples for omic analysis and electrophysiology. So this is the pathway that are involved. And the protocol we chose has been already published. So we, at the first day, we put the draw single dose, and then we wait until 2 days. We wash out, and we keep the cell until day 40. So the repetitive dose exposure, we put it 3-time dose. Every 2 days, change the medium and keep the cells until day 40. With the end that every day 2, day 6, we collect the sample for omic data and the sample for patch clamp. So this is the protocol that we use. The first part of this work has been already published, so I will not go too much in detail, but just to mention that the compound that we chose, just look, when we expose the compound with the cell to the compound, and we record 14 days, the effect, exposure in short time, you can really see compared to doxorubicin that is really toxic and with different concentration. That's the proof of concept. Then you can see here the arrhythmia, when you, according to the concentration, you reduce the beating activity of the cell, because the beating means that in the presence of compound, you reduce, you have some defect on ion channel or something else that will relate to the pathway. So if you remember this, protocol, we collect the cell at different time points. You have see here with this different cluster that has been from the omic, day 2 washout compared to day 6 washout. You have here the cluster of day 2, day 6, and day 14. You have washout scenario here. So it is really very clustering, and to show that in some, when we put the cell, we remove the compound, sometimes we have recovery from some gene or some pathway. And this, again, I will not go through. You can read the paper and to get all the information that is listed here. But what is curious here to see that day 2 washout compared to day 6 washout, day 2, that is gene that has been downregulated, and these are the genes that have been upregulated. We have clustering for washout, down, washout, day 2, and day 6 down. You see always we have some recovery gene that has been come back after washing out. But there are some that still really be downregulated. Of course, we take some, could not focus on all these genes. We take some genes that are involved in the functional, electrical function of the cells, and then we have to go through to the gene to identify those biomarkers that can be considered as biomarker. So, and they are listed there. So as I mentioned, we collect also the sample for electrophysiological patch clamp, and you can see here for those familiar with the patch clamp, manual patch clamp, do you know how difficult it is? You collect in each time point, we collect the cells, prepare the glass cover slip where we culture the cells, keep in the lab, and for each time point here, we have about 24 samples. So we have to collect these 24 samples every day. Means from Monday until Saturday, we are patient. So you cannot just imagine all this sample. You have to make triplicate experiment, at least. And for electrophysiology, you can just multiply this by 10, at least 10 experiment series to get. So anyhow, we use the manual patch cam because at that moment, we did not have really automatic patch cam that was really satisfying the criteria that we chose. So this is the manual patch cam, and those familiar with the system, you know, I still remember that every time student to me and say, this morning I cannot get the seal, so I don't know what happened. So we have to fix before starting experiment. You are not getting most of the time to concentrate on your data, but you spend most of the time to fix the setup. And this is manual patch cam. And there is no way you have to do it. Anyhow, we do it, and we classify those results in 3 categories, because the toxicity will mean you will have some effect on the action potential duration. It can be shorter, or it can be prolonged. It will be sign of arrhythmicity. So from this paper from Priori group really classify these 3 compounds. You have the action potential prolongation, maybe with terfenadine. You have this compound that's induced early after the polarization, and you have also this with really some kind of triggering. So the result that we collect from our experiment, we classify according to these 3 groups. So after collecting the cells, because you have to compare the 2 single exposure to day 6 3-time exposure to capture at least in the level if there are some cells that show arrhythmicity, with the idea that the cell, you have different subtypes. You have atrial, nodal, and ventricle. And why we do this is just because we realize that some compound will have effect on the Foley channel. They will block the Foley channel. If you block the Foley channel, means you have less nodal cells or not nodal cells at all in the patch clamp time. So that's why we select the cells and make some category that you can find here. So to come to that, you can see representative traces of those. Control, you have the action potential prolongation here. We have the early after depolarization will happen here. You have the triggery, means the calcium, it can be that the calcium canal open and they do not close, and the closing probability is taking so long time to return to the repolarization phase, or it can be that you have more activation of the calcium coming from sarcoplasmic reticulum that will trigger the cells. And of course, you have some category that we call it unspecific category. So you can see here how is the representative distribution of the cells. And of course, you have the action potential, the calcium current that also affected. You have also the natrium, the sodium channel affected through that. But anyhow, we show that with the doxorubicin, we can really capture the early, combine the IPS with omic data and electrophysiology to capture the early effect on the cells, and to understand the molecular mechanisms on the live multimodal that typically involve a multi-gene, because it is the combination of different genes. So it is really difficult to know which gene, if this gene is going down, maybe there are some compensatory mechanisms happen in the level of cells to cover the event. So it is really difficult to manage that. The provocative question is, does automatic patch clamp system have the potential to replace manual patch clamp system in ion channel research today? What are the challenges? The CO mentioned the challenge. One of the challenges that are always, as electrophysiology for a long time, it is to capture the event from the first time point of exposure until maybe day 2, or day 3, or day 4. Means long-term electrophysiology recording from ion channel in the presence of some compound. That is the challenge. So I will end to thank all my colleagues involved in this activity, the former member, and those guys always taking time to collect this electrophysiology data. And thank you for your attention.
Okay, thank you very much. So we started a little bit late, so I think we've got time for one question, and if not, you can always grab Filomain and coffee. Yes, in the middle there. Can we have a microphone just in the middle?
Lovely, thanks very much. I just wondered whether you tried this on other cardiac cell types rather than just cardiomyocytes?
Yes, but as you know, it's not easy to do it with manual patch clamp. So we have also disease models, and we have neuron cell that we also generate in the lab, and other cardiac subtype that happen with just 3 nodal, atrial, and ventricular cells, not more.
Yeah. Okay, thank you very much.